Hi everyone, Professor Davis here, and I'm going to do a brief review with you for your lab final. So we're going to go through each of the labs that you've completed through this semester, and we're going to kind of hit the high points of things you need to remember or know about them. So in lab number 1, this one focused in on microscopes. So microscopes are used to magnify objects that are too small to see with the naked eye. So we have to use an instrument such as a microscope to help us see these really small things. Now there are different types of microscopes. The microscope that this lab focused in on was the light compound microscope, and it's the one most of the time that we would use in our labs. It can magnify things 1,000 times larger than they are. But electron microscopes are a lot larger, and they can actually magnify things to huge amounts. We're talking about like anywhere between 30,000 and 50,000 times their normal size. And so this is going to be a more advanced type of microscope. Now, parts of the microscope that you need to make sure that you know. The ocular part of the microscope is the eyepiece that you look through. The nose piece is going to house the objectives, which are the magnifiers, and they rotate so you can use the different ones. We also have a stage where the slide is placed. It holds the slide in place, and it can be moved up and down or left and right. The diaphragm is going to be where we adjust the light, and we can do this by moving a little lever that opens or closes the iris. There are 2 focus knobs that are going to adjust the specimen into focus for you. The larger one is called the coarse focus knob. It gives you a rapid and approximate focus, whereas the fine focus knob is a lot slower, but it's more exact in its focusing. The objectives are the magnifiers. They're going to make it where you can see the object better. These are going to be scanning, which is the lowest magnification, followed by low, high, and then oil being the highest magnification. All right, looking at lab number 2. Lab number 2 focused in on the scientific method. So we have a sequence here of the scientific method you need to make sure that you know, and you need to know it in order. So we have an observation followed by a hypothesis. We then do an experiment, which leads us to our conclusion. Now, when we develop experiments, good experiments are going to have different variables present. So the independent or experimental variable is what's being changed, or what we are going to be able to modify in order to see what happens. That's the independent or experimental variable. Determining and actually measuring what happens is called the dependent or responding variable. It's what changes due to the experimental variable. It responds to what you altered. Now, in a really good experiment, we're also going to have a control group. Control groups go through all the steps of the experiment except the experimental variable. This is to help us determine that the experimental variable is the cause of the change in the responding variable. Now, the hypothesis. Guys, this is known as a tentative explanation. Sometimes it also gets termed as an educated guess. The key thing here, though, is that it is educated, meaning you do have some knowledge about it when you make that statement for your hypothesis. All right, moving on to lab number 3. Lab 3 was focusing in on chemistry. And so with chemistry, we were talking about some macromolecules. You need to make sure that you know the building blocks of these macromolecules. This includes carbohydrates, which are built by monosaccharides, lipids, which are composed or built from fatty acids, proteins come from amino acids, and nucleic acids are built using nucleotides. Make sure that you know these and you can match them appropriately to their building block to the macromolecule. We also see some characteristics of lipids. The main characteristic you need to know about fats or lipids is the fact that they are hydrophobic, meaning they do not like water. So they are not going to dissolve or be soluble in water. Now, in these experiments that you read about, and you actually performed one in lab number 3, there was a control that was used in the experiment. And this control is the one who gave you a negative test no matter if you were testing a carbohydrate, lipid, protein, or nucleic acid, because it does not contain any of these things. This control is water. Water is one of the best controls to use here. We also need to know the reagent that was used for each test. Now, when you guys did this lab, for simple sugars and proteins, you just read the nutritional labels and told us whether it was positive or negative. However, in the lab, it talked about the reagents used. So you are going to be held accountable for these. The simple sugars use the reagent called Benedict's. Benedict's goes from a blue color to an orange or red color if a simple sugar is present. On the other hand, for proteins, they use the Biuret. Biuret also starts out blue, but it changes to purple if proteins are present. Now the middle one here, the complex sugar, you did do the iodine tests. Iodine starts out as yellow, but if a complex sugar, like a complex carbohydrate is present, it changes to a blackish purple color. And you guys saw this in the lab. All right, go into lab number 4. Lab 4 is going to be looking at cells. So we have prokaryotic versus eukaryotic cells. Remember that prokaryotic cells are very simple cells. They do not contain a nucleus and they do not contain any organelles except for ribosomes. These are going to be examples like bacteria. Eukaryotic cells, on the other hand, are a lot more complex. They do contain a nucleus as well as lots of organelles like mitochondria, rough ER, smooth ER, Golgi. These guys are the more complex cells, so these are cells that make up plants and animals. Now, we do also need to be able to ID the difference between a plant cell and an animal cell. When we look at plant cells have a couple of structures that animal cells do not. The first one is a cell wall. They also contain chloroplasts because they do photosynthesis. And they have a very large central vacuole which helps them store water and nutrients. On the other hand, animal cells do not have these three structures, but animal cells do contain centrioles, which help with cell division, and they contain lysosomes, which contain digestive enzymes and help clean up the cells. Another thing you need to know is you need to know what happens to a cell if it's placed in a hypotonic or a hypertonic solution. So if a cell is placed in a hypotonic solution, it swells. And so you can kind of think of it as hypotonic. It gets bigger. Now with these hypotonic solutions, if it's an animal cell, specifically a red blood cell, we call this hemolysis because what happens here is that it swells until it bursts because there's no cell wall to stop it. And lysis means to break or burst. On the other hand, in a plant cell, we call it turgor pressure because the cell wall is present and it puts pressure on the cell wall, but it does not burst. In a hypertonic solution, the cells shrink, they shrivel up. So in animal cells, we call this crenation because the cells shrivel up and they get kind of spiky, kind of look like stickers. In plant cells, we call this plasmolysis because the plasma membrane pulls away from the cell wall. And so that lysis means it breaks away from the cell wall. Two other terms you need to make sure you know is tonicity and osmosis. So tonicity is the relative concentration of solute in a solution. So a solution might be hypotonic if it doesn't have very much solute in it versus a hypertonic solution that has lots of solute. So an example would be distilled water versus salt water. Okay, there's a big difference there. All right, so that's what we look at for tonicity. Another one is osmosis. Osmosis is the movement of water from high concentration to low concentration. Water likes to spread out from high to low, and that movement is called osmosis. All right, looking at lab number 5. Lab number 5 focuses in on enzymes. So when we're looking at enzymes, you need to be able to define an enzyme as well as the substrate. Guys, an enzyme is an organic catalyst. Normally it's a protein. And when we say it's a catalyst, it means it speeds up the chemical reaction. And it does this by lowering the amount of energy it takes to start the reaction. This is what makes it go faster. Now, what goes into this reaction and what the enzyme actually works on is called the substrate. This is the compound the enzyme acts on, and we call this the reactant. Enzymes, a lot of times, not always, but a lot of times enzymes can be identified because of their name. And this is because in their name, they tend to end in ACE, A-C-E. This would be things like catalase, sucrase, lactase. These are all enzymes because they end in ACE. In this lab, you did look and investigate 3 factors that affect enzyme activity. These included concentration of the enzyme or the substrate. So that's the amount of enzyme or substrate that was added in that determined how it was going to react. We also looked at temperature. We had it where it was hot, cold, and room temperature, so temperature. And then also pH, and pH is whether it was an acid or a base. So these three things were factors that affected how the enzyme worked. Now sometimes, guys, especially in temperature, if the enzyme gets disrupted enough, it starts to unfold, and we say that it's been denatured. This is when an enzyme loses its shape. It's not folded properly anymore, and this means it can no longer do its job. So in other words, it's no longer functional. It doesn't work. All right, we're going to look at labs 6 and 7 together here because this is dealing with mitosis and meiosis. Now with mitosis and meiosis, you do need to be able to identify the phases of both and what happens. But let's do a brief overview of the difference between mitosis and meiosis. Remember, mitosis is one division, so the cell divides from 1 to 2. Two identical daughter cells are created and they look exactly like the parent cell. And since this has just one parent cell, this is a type of asexual reproduction. We use this for growth and repair. But in meiosis, we see that there are two divisions that take place, and this creates 4 unique cells that have half the number of chromosomes. This is so that we can create things like egg and sperm, so that when an egg and sperm come together, we can then create a whole new organism. So meiosis is part of sexual reproduction. You need to also be able to ID the phases of mitosis and meiosis through pictures. So you need to go back and review those labs with those pictures. And I'm going to show you a few on the next slide just to kind of go over them. We also see for cytokinesis in plants and animals, there's a difference. And the main reason is because of the presence of the cell wall in plants. Since they have a cell wall, they have to build a cell plate when they divide into 2. Whereas animal cells do not, and they create what we call that cleavage furrow. The last thing you need to be able to do here in this lab is where you calculate chromosomes if they are diploid or haploid. Because remember, diploid means we have 2 sets of chromosomes. Haploid means I have half. So if we look here, diploid organism may have 24 chromosomes. How can I calculate the haploid cells? Well, I would take half of 24. So half of 24 is 12. So the diploid is 24, the haploid is 12. On the other hand, let's just say my haploid cell has 10. How would I determine the diploid number? Well, I would take that 10 and I would multiply it by 2. So 10 times 2 is 20. So the diploid number would be 20, the haploid number would be 10. Now guys, when I ask you to calculate these, they'll be simple numbers. You will not need to use a calculator on them. All right, so let's look at some pictures and see what stage we're looking at, whether it's mitosis, meiosis, or if we're looking at things like prophase, metaphase, anaphase, or telophase. So we're going to kind of look at each of these stages. So in this first one, we see that the chromosomes are being pulled apart, but since the chromosomes are being pulled apart and the number hasn't been halved, because most of the time in our pictures, we start with 4 chromosomes, we see here that this is going to be anaphase of mitosis. So anaphase of mitosis. In this picture, though, we have something called tetrads. When they come together and they are combined where there are 4 chromosomes lined up together, this is going to be tetrads, and this is only found in meiosis 1. Since they're lined up in the middle of the cell, this is metaphase, and so this is metaphase of meiosis 1. In this case, we see that the chromosomes are not lined up in any special way. There are 4 of them, but the nucleus is going through the process of fragmenting. In this particular case, we see that this is going to be prophase, but this is prophase of mitosis. All right, so we've already hit on one of these. When the chromosomes are being pulled apart, this is known as anaphase. So since the chromosomes are pulled apart in anaphase, we do see that these are still tetrads. So this was meiosis 1 of anaphase. Sorry, this is anaphase of meiosis 1. Here we have the chromosomes lined up in the tetrads, but the actual nucleus is fragmenting. So this is prophase 1, so prophase of meiosis 1. All right, here's a different phase. We see that they have now divided and we're going through cytokinesis. The nucleus is reappearing, so this is telophase. In this particular case, this is telophase of mitosis. All right, so we just said this was telophase, but what's different here? Well, here I still have those chromosomes that are present. This is going to be telophase 1 of meiosis 1. So telophase 1, meiosis 1. All right, staying along with telophase. Again, we know this is telophase, but how many cells am I going to end up with? Oh, I'm going to end up with 4 cells. So this is the first one that you see that is meiosis 2. So this is telophase 2 of meiosis 2. Here I have 2 cells. They're about to get ready to divide again. The nucleus is fragmenting, so this is prophase. So since I have 2 cells going through this, this is prophase 2. All right, so this just gives you an idea of going through pictures, but make sure you do review them from your lab book. All right, now let's look at lab 8 and 9. So when we're looking at lab 8 and 9, we do need to look at some definitions because this is genetics, and you'll notice that genetics had lots of definitions we needed to know. So what is a karyotype? A karyotype displays an organism's chromosomes, and it does it in an orderly fashion, and we should have 2 chromosomes of each. So 2 number 1s, 2 number 2s, 2 number 13s, 2 number 21s. That's what we should see in a karyotype. Pedigrees, on the other hand, are used to determine the pattern of genetic inheritance along family members. This is where it creates kind of that family tree with those circles and squares. We see that we can determine certain kinds of disorders using a pedigree, things like autosomal dominant, autosomal recessive, or X-linked recessive. This brings us to some terms, heterozygous versus homozygous. Hetero means different. So heterozygous is where our genetics have the 2 different alleles. So there's a capital letter and a lowercase letter, like big T, little t. We could use big A, big B, little b, but that's heterozygous. Homozygous, on the other hand, is going to be where they're the same. Homo means same. So big T, or little t, little t. We could also use big A, or little a, little a. Now, what determines if it's a big letter or a little letter? Well, that is determined if it's dominant or recessive. Dominant means it's the capital letter, and it can be expressed where we actually see the dominant trait if there's at least 1 big letter. So big T, or big T, little t would show the dominant trait. The recessive trait is the little letter, and we only see that expressed if there are 2 little letters, little t, little t. All right, now this brings us to genotype and phenotype. Genotype, guys, is the genetic makeup of the individual. These are those letters we just talked about. So like big T, little b, that kind of thing. Whereas the phenotype is their physical appearance, how we would explain them. So phenotype would be something like they're tall and they have blue eyes. Some disorders that we did cover that you want to make sure that you review. Turner syndrome is a monosomy, and in Turner syndrome, remember they have 1 X and they're missing the other. The O here tells you it's missing. This is a female, but they only have 1 X, and we would be able to see this on a karyotype. Klinefelters could also be seen on a karyotype, and this is where we would see XXY. They have 2 X's, 1 Y. This is known as a trisomy. Another disorder that we talked about are X-linked characteristics. These X-linked characteristics are found on the X chromosome, and they tend to affect males more often than females because males are XY, whereas females have an extra X copy to help counteract this. So they affect males more often, and they often do skip a generation when we look at the pedigrees. All right, last but not least, let's look at lab number 10. When we're looking at lab number 10, we do have the codon. The codon consists of a number of nucleotides, and this number is 3. So they do have 3 nucleotides, and those nucleotides code for 1 amino acid. So make sure you know a codon has 3 nucleotides, and it codes for 1 amino acid. We also need to know your base pairing rules. When we look at these rules, we want to go from DNA to DNA, and then we want to look at the rules from DNA to RNA. If I'm going to do my base pairing from DNA to DNA, A always pairs with T and G always pairs with C. So one way to remember this is the straight letters A and T go together and the curvy letters G and C go together. Now that rule doesn't really work when we go from DNA to RNA, because when we go from DNA to RNA, there is no T in RNA. So if I have an A in the DNA, it's going to pair with U, okay, because T is not in RNA. But if I have a T in the DNA, it still pairs with A, and G always pairs with C. So what do I use as a template to make the messenger RNA? We use the DNA strand. So the DNA is going to have the blueprints or the instructions on how to make the messenger RNA or mRNA. This brings us to two terms, transcription and translation. Transcription, guys, is where I take that DNA and I'm going to copy and make a copy of messenger RNA. Now, guys, since this is DNA and RNA are both nucleotides, I'm transcribing. I'm just copying down the message. However, in translation, we are going to go from an mRNA strand, RNA, which is a nucleotide, and we're going to change the language to an amino acid sequence. We're changing it to a protein. Since these are two different things, this is why we call it translation. This would be like if I was translating English to Spanish. They're different. It's going to look and sound different, and we have the same thing here in translation. We're converting a messenger RNA strand into an amino acid sequence. All right, so that reviews our labs 1 through 10. If you have any questions, please let me know, and good luck on your final exam.